All right, welcome to TIL Talks with Chris White, where we talk TILs, tumor-infiltrating lymphocytes, and all advanced cell and gene therapies, both with patients and caregivers, along with industry leaders. Let's get going. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. What's up? What's up? Welcome back to another episode of TIL Talks. Today, I have my repeating guest, Dr. John Patterly from Carolina BioOncology Institute and BioCytics. This is, I believe, now our 10th recording. Super excited. Man, give us a little 101 on what's been going on and where we're going to pick up from where we left off.
Yeah, good, Chris, to see you again. Forgot we've done so many. Yeah, it's been almost months worth of good banter back and forth. So I think the news that made the headlines this week, I'm sure a lot of your audience saw it, was a research trial results at ASCO, American Society of Clinical Oncology, which is always the end of May. It makes
viral.
NBC News. So that was one of the drugs that we have been doing in phase 1 trials, actually. So it's a drug for pancreatic cancer targeting KRAS G12D, or in fact, all the KRASs. So it's a pan-KRAS inhibitor. And we've had that study open, or the predecessor to that study, open the past 2 years. And the study they presented was a big phase 3 randomization, but we had done the earlier phases for that and another drug with it in combo. And I tell you, our other doctor here, Dr. Gandhi, just presented for our whole research team in our clinic at our huddle where we talk about the patients, and we took 20 minutes aside to show the ASCO plenary session, and there was a standing ovation.
I was going to say,
standing ovation among these nerdy scientists, because the survival curve was so big, and the difference in versus study drug was such a big difference. Great news.
It's everywhere.
It's funny on that ASCO thing. I'm like, I have to try hard not to talk about the specific drug, but just that it's used and they can look it up.
I'm really glad that you brought that up, because I saw multiple people on multiple platforms that were in the audience that all made posts about it, standing up, major reviews, the opening plenary, huge. I only been to ASCO once, and that was in 2024.
Yeah.
But I think I saw somebody post something that this year cell therapies and clinical trials is making a huge shift and is a big focus at ASCO this year.
Yeah, Dr. Gandhi, that good news update about the pancreatic clinical trial. We just saw the plenary recorded session here in our clinic because Dr. Gandhi, our other oncologist, and some of our other research team went, and they were telling me that the excitement, the audience, the nerdy oncologists and all the research, they were standing up, clapping, standing ovation. And the ovation went on for a good 45 seconds, which is very atypical for the ASCO community. But basically never has there been any former pancreatic cancer trial ever to show that big of an improvement in the doubling of the overall survival. So the magnitude of the good news with this RAS inhibitor, pan-RAS, meaning it targets all the RAS mutations. So that was just great. And it's only a pill. So it's just a pill, once a day pill versus old-fashioned 1980s chemo. The pill won in a big way. Progression-free survival, overall survival. So definitely the pill has a future. What they also showed that patients' overall side effects, the different toxicities, and the chemo is pretty generally toxic, low blood counts and all that. The pill has some rash. It's mild to moderate rash in probably 80% of the patients. But the rash was treatable and manageable. Holding and reducing the dose, the rash would improve. But I think the end result is all the investigators that commented on that trial say that it's going to become the standard of care for pancreatic cancer.
Huge.
Yeah, huge. And they're going to be combining that pill with other tumor types here very soon, gastric, colon, other cancers, biliary cancer. So it's exciting to see a small molecule, a targeted, it's a driver mutation, these KRAS G12D, G12V, G13, that whole class of KRAS mutations. It seems like this pill seems to hit and target and works good. So yes, that is exciting for the research community, and hopefully the FDA will get it approved approved as commercially available in the next weeks to months. In the meantime, many of the research clinics, including our research clinic, we are opening it as what's called not really compassionate use, but it's called expanded access.
I was
So
going to
it is
ask
better
that.
than compassionate use. Expanded access means that it's more readily available. You just have to come to the clinic. Technically, it is a trial. It's like a very thin trial just to get access and to enroll and then get it dispensed 1 month at a time of the study drug. And we work with your local oncologist to make sure your symptoms and side effects are being managed and coach you through that expanded access trial. EAP, expanded access trial.
And is that an inclusion exclusion criteria that is posted?
It's pretty broad. Yeah, I mean, it's so broad that it's like if you have stage 4 pancreatic cancer and you have had any prior treatment, you can get access to the pill as long as you can walk in the door. I mean, that's pretty
So
much
there's
as
no
long as you're
like
safe enough.
limitations on your progression or
I think the way it's written, going by memory here, most of these expanded access program, EAP, say that you have to have failed something, you have to meet
second
very
line of
broad
treatment at least.
eligibility, like be good enough performance status that you can get out of bed and make it to the clinic safely, to follow instructions and have informed consent and have generally safe laboratory values. I mean, if somebody's in kidney failure, we can't do it, or somebody's in complete malnourished and not safe to walk in the door, then
Yeah,
they
that would be a
eligible.
risky.
But if you have a good support system and you can follow instructions and you're motivated, I think there's going to be at least one research site in most major cities. I think for the North Carolina area, we're for the expanded access program. So any
That's
of your
awesome.
audience can call, and we already have got patients that are on the list. And so that's it has to go through RB approval. It's still a trial. It's opening up, but it's opening up literally in the next few days to a week. So we'll have that available very soon.
Very cool news, man.
Yeah. So that's good. And so it's not immunotherapy, it's not cell therapy, but it's a pill and it works. And so it's good news for pancreatic cancer patients who've never had any good options like this that were so targeted.
It's incredible. Like you said, a 45-second standing O.
Yeah.
over there.
On LinkedIn, you'd get to see the docs like going around taking
LinkedIn, Instagram, whatever, dude. Facebook, it's everywhere. Like you said, the national news.
But this one's not just hype. Like sometimes things are overhyped around ASCO each year, but this one's the real deal. It really is a big impact. I mean, there's never been a big shift in standard of care for pancreatic. Unfortunately,
when there's limited options to begin with, it's a huge shift for those patients.
Historically, pancreatic cancer is so bad that the incidence rate pretty much equals and of all stages equals the survival curve. It's kind of like so there are no like stage 1, 2, 3 patients that really, unfortunately, very few survive. The number of cases there are ends up being the number who died the next year.
Yeah,
that's how bad of reputation. We tell patients some cancers they're so stubborn, they have a bad reputation because they're born out of inflammation. In pancreatic cancer, it's such a digestive enzymatic organ, and pancreas has no tissue plane. There's weird things about the pancreas itself, and it's so wrapped around so much digestive function, and the enzymes within the pancreas just digest more, and then there's inflammation, and tumors know how to hide with the inflammation in the pancreas. So it has a lot of, and there's no real good symptoms that predict it. So there's many biological reasons why pancreatic cancer gets its bad reputation.
There's a lot of microbiome.
Maybe their patients are malnourished, they're often smokers, but it's crazy. We're seeing in North Carolina, even non-smokers in their 40s and 50s are now coming in the door with pancreatic cancer. Historically, it was just the heavy smokers in their 60s and 70s, but now younger
patients used to be the most contributed cause or
smoking was a big swallowing the tar when you smoke. If you don't inhale it, then you swallow the tar because it's in your mucus and when you swallow, it's in your stomach, it's in your bile duct, it's in your pancreas. And so that was the mechanism why they thought pancreatic cancer was related to tobacco use for many years. But now we don't know that in the younger patients getting it, we don't know it's probably some industrial toxin that
environmental factors, food. I would definitely say probably something with the food. I mean,
some of the inheritable genes might have a little bit of a increase in pancreatic cancer in some of the hypermutator like BRCA mutants. And so in those, if not women that get breast or ovarian, it may mean they have a slightly increased risk in some other types of cancers, including colon or pancreas. But anyways, it's good news. I just wanted to make sure we talk about That's the happening this week.
I really wanted to go this year, but things happened and wasn't able to make it. But man, it would have been really cool to be in that room while that was going down.
Yeah. So that company has a lot of other drugs in the pipeline, and they had a slide at ASCO. There's probably about 40 to 50 other KRAS targeting drugs in their pipeline. So what that means is there's going to be a lot of other pan-RAS or very specific targeting RAS drugs. They may ultimately in the future be used in combo or sequentially. Like for lung cancers, you have like brain. Some lung cancer small molecules targeting EGFR will go to the brain, and the drugs, some cross the brain, some are improved second and third line. The more mutations you get, the more you have to switch to different small molecules. So I can see that happening with KRAS. Pretty soon, it's like right now it's a pan KRAS, but then next year to be like, oh, what KRAS mutation do you have? Oh, well, there's a new drug coming out for that one, and this one, KRAS G12D, KRAS G12V. So we'll probably start seeing more cocktails of these KRAS drugs and other doublets, or how to combine or give them in sequence. So
it's also just another reminder to highlight how important genetic testing is for your mutations. And
yeah, they can now in the bloodstream, they can detect a lot of these mutations right in the bloodstream. So even if you can't get a good pancreas tumor biopsy, pancreas is a tough organ to biopsy, by the way, because it's not an easy aim. You got to have a pretty invasive endoscopy to get it, or so it can be where some of the mutant drivers, the KRAS mutations, can be detected in the bloodstream. Ideally, a biopsy is the gold standard, but it still can be detected at what is your genotype, your mutation driver that can be detected from the blood.
now, can that be found within just like your CBC blood panel, or would this
No,
have to be more specific, like a Signatera type of thing?
Yeah,
Or
it'd be one of those next generation sequencing type of platforms where they can discover a strand of DNA. And the way the DNA has, they say like epigenetic mutations, like lack of methylation or aberrant methylation. There's other signatures in the bloodstream that you just have to have highly sensitive genomic assays can test for it. So there's about a dozen or so labs that do that, and they all compete very good. So your local oncologist would know how to get the peripheral blood tests for the KRAS gene mutants testing if you can't get a biopsy, or if you had a biopsy that was too small to do genotyping on. So yeah, but good. Well, yeah, so we should pick up where we left off last time. Last time we were talking, we got about halfway through. So yeah, the last time I presented some slides that I gave at a community oncology meeting north in South Carolina, down towards the beach. And it was basically trying to educate oncologists. Why should they build a phase 1 research program? Why does this matter? And we talked about the whys here. This is kind of where I left off. Like, oh, there's 9,000 new drugs. It's a big pipeline, but only 2 to 3% of cancer patients participate. So we need more investigators. So this presentation was a call out to the investigators or to the oncologists in the audience to say, hey, why don't you consider learning how to be an investigator and do clinical trials and take on the duties of an investigator? So one of the teaching points is, well, what are the deliverables if you are an investigator and you're contracting with biopharma industry? And these are the 3 main deliverables that the biopharma industry expects from an investigator who's doing research trials in their practice. The first is they want professional services, meaning they want to know that you're a good doc, take good care of patients, you're giving good care. You've got an in-house lab, you can do blood counts, you've got a pharmacy, you've got nurses, an infusion room, you can have a team,
whole
take calls with
infrastructure.
you, like all that infrastructure. Yeah, professional service. The next is they want to know that you can enter data. So underlined there, it's like high quality, accurate, verifiable data in a timely entry, so that as you are running the trial and talking to the patient to get the side effects and grading them and scaling them and attributing them to the different causes of their side effects, that is going within 24 to 48 hours, it's going into a database. And that's the other deliverable. So can't fall behind on the data, because then the drug company can't keep up with how the trial is going. So you have to have dedicated data entry coordinators. That's what they do. At our clinic, our coordinators kind of do both. They'll coordinate the trial, talk to the sponsor, talk to the family, and then go back. And in the afternoons, they're entering the data. So we keep it kind of focused. A coordinator for each trial enters the same data. And then the third deliverable, that if you want to become an investigator, is the sponsors expect you to be engaged with them. Engaged meaning you're giving them not your professional services of the medical care for patients, but you're giving them your professional services focused at them for what they need, which is they want to know your opinion about, does the drug side effects, are they manageable? Does the drug have anti-tumor activity? How are you managing the side effects? Dear Dr. Powderly, how do you think we should coach other doctors how to manage the rash, for example, or the diarrhea, or the indigestion? They also want feedback on how to edit the protocol to make the protocol more streamlined, maybe to add some safety things in, maybe to take away some unnecessary hurdles. So, and they want you to be on the phone calls. So they have these investigator calls, like these days it's like every other week, usually on a Thursday or Friday. As the clinic winds down, they have like 30-minute calls where the investigators from all across the time zones will talk about how the trial is going, how side effects are coming on. And then they want you to be on the calls for the safety monitoring committees, which is the big decision if you decide to escalate the dose or not escalate the dose or expand the dose. And then when they do get interim data results, like from the first 50 patients or so, they might look at the aggregate data and help get investigator feedback to the biopharma sponsor about the aggregate data. Like, does the survival curve look good? Does the toxicity look bad? How do we assess those things from our professional opinion? And the PK/PD data, that's pharmacokinetics and pharmacodynamics. That is, how does a drug in the body get excreted, metabolized? How long does it last? And then the dynamics are, what does it do to the body? What does the drug hit the target? Does the drug actually affect the biology of the target? And we can have measurements for both of those things with the many blood draws. The patterns of the toxicity. And then, of course, they also want the docs to give their input and feedback on abstracts and publications and manuscripts about the trial. Like this ASCO, we just talked a little bit about ASCO. I mean, all the 10 slides and all the abstract data, that's all been reviewed by the investigators who sign off on it to be listed as an author. So those are some of the duties as an investigator. So next is, this is a good one for the patients, and some of the docs are learning this, or they were taught it many years ago. But so interestingly, in phase 1 trials, where the purpose is to figure out what's the right dose, you got to start somewhere. And so there is actually mathematical equations about, where do we start? How low do we go? What do we pick as our low starting dose in order to begin the phase 1A trial? Because it's the very first dose in a patient.
I say you just can't throw out some random dosage and just see what happens. You have to.
So this is it. This is how we figure out the first dose, as this is pretty much the guidance from the FDA and the other regulated industries of what's the traditional standard. Usually they'll start the lowest dose about 2 or 3 log orders below what was the toxic dose in a mouse. Okay. So once you go 2 or 3 log orders below that, which is really, really, really low, they take that number and then they will add it to that number based on what is called a Fibonacci number. And a Fibonacci number is, if you take a number and add to it, it will increase as you add to it and add to it. And this is like something interesting about nature. Like this, a lot of things in nature have these spirals and curves. And so down in the lower right, I'm showing you the Fibonacci number, the mathematical equations of how like a nautilus shell, for example, would expand its curves, and the diameter of each of those curves expands based on 1 + 1 = 2. And you can see it starts with 2. 2 + 1 = 3. And you see down in there, and then 3 + 2 = 5. Okay, now then 5 + 3 = 8. Then 8 + 5 = 13, and then 21 + 13 = 34. So you can see, as the nautilus shell, how it goes around as a very natural, biological, mathematical Fibonacci equation. That is how we decide about how we're going to dose escalate. In the incremental ratio, or the slope of that curve around the nautilus shell, is a factor, or I would say a constant, if you will, a factor that can be used to make the slope a little bit higher, quicker, or a little bit more slower, more prudent. So you can be a little more aggressive or a little bit more prudent. So in doing these phase 1 studies, when would we want to be really prudent? Like kind of a slow escalation might be a first-in-class drug. Like the first time that entire class is ever given to any human. Now that one, you'd want to be real prudent, just go up a 33% increment of your Fibonacci numbers. However, if it's like the 20th time that class has a new drug, like a copycat drug from another sponsor, another competitor, say, oh, we know this class. This class has been around a few years. We can escalate it a little bit quicker. We can get through some side effects quicker, dose finding quicker. So that is where we get how we escalate doses based on these Fibonacci numbers. And I thought that was interesting for your audience to hear that. It's not just guessing. There actually is some good science about that.
I was going to say, for safety and efficacy, you got to have something like this to build upon.
Yeah, and that's exactly why they got to start with a small number of patients, give the patients about a month, usually 2 to 3 weeks, before you enroll more patients in the next dose level. But the next dose level, you go up that 33 to 100%, depending on the slope of the Nautilus Fibonacci number equation. So speaking of that, so here's the next slide. So these are the two ways on the left and right on how we can escalate the dose based on the Fibonacci equation. So before we showed you the equation, now we have to pick how many patients at a time can we give that dose level to, okay? So on the left is the traditional historical, we call it the 3+3 design, which is you give the drug to 3 patients and you wait a month and then you see if those patients had any side effects, and if they tolerated it and there was no bad side effects, then you can go up to the next dose level on the next 3 patients. So at the bottom there, it shows 3 dots or 3 patients. The green dots, oh, they did well, no side effects, okay? At the starting dose, 3 patients, wait a month, go up 3 more patients, next dose level, 3 more green patients, all did well, no side effects. Then the next month you can bring in 3 more patients into the phase 1. This is global, this is worldwide, global, like that means 1 patient on the East Coast, 1 on the West Coast, maybe 1 on Europe gets a slot. Then the next group, 2 patients tolerating it, but the 3rd patient is in red because they had a side effect. And not just a mild side effect, but a side effect we would say is a DLT, or a dose-limiting toxicity.
Okay.
And a DLT means the drug had such a bad side effect that the toxicity, we had to hold the drug or manage the patient a little more aggressively because the side effect was moderate to severe. And an example of that would be like, so that's how we describe side effects. A mild side effect means you get out of bed, do your chores, eat your meals. It may bother you a little bit. That's a mild side effect. A moderate side effect means that you're getting out of bed slow, eating half meals, doing half your chores. You need some treatment.
Becomes debilitating.
Yeah. You might need to hold the drug. You might need some supportive like IV fluids, nausea medicines. We have to start writing prescriptions. That's moderate. Moderate is still allowed to continue that dose level. But a severe, which is usually a dose-limiting toxicity, is severe, means you had to stop the drug because the patient had a severe side effect, like a really bad rash on most of their body, or more than 6 to 10 stools a day, diarrhea. Really bad, didn't eat food for like 2 or 3 days, or God forbid, ended up in the hospital. That would be an automatic red dot, stop that dose level at that moment. You've got to tell the drug company, who then tells the FDA, and the investigators are aware and say, okay, we declared 1 DLT, 1 dose-limiting toxicity. Now, do you stop at 1 DLT? Depends. If it was a DLT that resolved pretty quick, like within 48 hours, and the patient recovered, and then you can ask permission to restart that patient, that 1 red dot, at the lower dose level, and the sponsor might allow that. Then you're still left with the decision, are we forced to expand, or are we forced to go down? And usually if there's only 1 DLT, 1 dose-limiting toxicity, among the first 3 patients in a phase 1 trial, and if the patient recovers and does fine after they recover, stopping the drug, and all the investigators agree and the sponsor agrees, and the drug otherwise looks promising, they will allow you to do 3 more patients at that dose level. All right. So, because if you have more than 33% DLT, then you got to stop. But less than 33% or less than 1 in 3, you're allowed to stay on. Equal to 1 in 3, you can stay on. So now that you have six patients in that cohort, 1 of 6 green dots means you cleared the cohort, but you're cautious because you had 1 dose-limiting toxicity in 1 patient. So then you're allowed to go to the next dose cohort. Okay. And in that next cohort, you say, oh, the green patient, first patient did well, 4 weeks go by, but the second and third patient, bad news. They both had a severe toxicity. And guess what? Usually it's the same side effect that the lower dose. It's usually the same thing happening.
Yeah.
Bad diarrhea or a bad rash. And so now the investigator community and the sponsor say, okay, now we're kind of hitting a wall, a DLT wall, a dose-limiting toxicity wall. We don't want to go any higher because now the drug's starting to show it's too toxic. So then what they'll do is they usually will go back down to that dose level where there was 1, 1, and 6, right? And they'll expand it and they'll say, okay, now we're moving into dose expansion phase. Okay, not dose escalation phase. We're going into dose expansion phase. And then that's when historically they would often move it into phase 2, or they would try to move it into comparing the dose level 3 and the dose level 2, just getting some more data. Nowadays, they might try to get an in-between dose and say, hey, can we go halfway between the DLT and the 1 of 6? Can we try to find an in-between? They will try to choose an intermediate dose, which is allowed. And those are the decisions that's our week-to-week decisions. We work with sponsors trying to figure that out. So that left is the traditional 3+3 dose-limiting toxicity trial design, which was, I would say that was the predominant way to do drugs for the past 20 to 30 years. Now, on the right, there's a little more of a more modern way that we're seeing how to do dose escalation, which I'd say it's starting to take more foothold. And the FDA blesses both types. The one on the right says, hey, we don't have to start with 3 patients. We can start with only 1 patient. And it says we can start with, and if 1 patient at the very bottom lower left, if he tolerates that first dose and he does fine, let's go to the next group and dose 2 patients. And if those 2 patients are doing it well, we can then go to the next number of patients. So then this way, we're kind of doing it not just 1 out of 3, we're doing it a little bit more optimized, optimal interval, where we can basically go through this grid on the lower right and try to, as we increase the number of patients evaluated, we can determine based on each group, can we go up higher or can we have to de-escalate or stay the same or eliminate the dose level. So the one on the right is a little more interactive, a little more dynamic, a little more phone calls and engagement with the sponsor and the investigator team. But the one on the right lets us be able to also get to the right dose level based on the same approximation out of 1 out of 3 to 2 out of 6, 33 to 33% range to try to figure out what's the right dose level within that grid on the right, if that makes sense.
Yeah, absolutely.
So I just wanted you guys to know that. So then a couple other things that are been changing. So the historical way we used to do phase 1s in the US was even if you hit your DLT and you said, okay, I went too high, I'm going to back down and then try to figure out 2 dose levels. They usually would go back down to a medium dose level below the DLT and then a lower dose level, and they would try to dose optimize between those 2 levels. And they would try to figure out that dose optimization while they were still in phase 1 and 2. That worked fine for many decades when the drugs in development were chemotherapy and they were pretty toxic. Because in chemotherapy drugs, you could dose optimize during a phase 1 and phase 2 because the toxicity would mirror the dose level. They were in the next slide kind of explains that. Like here on the left, if you look at the red toxicity and the blue efficacy as to how we would determine if a drug is too toxic, that usually reflected the efficacy right along the same way. And you experienced this, Chris, because you had chemotherapy. So chemotherapy toxicity almost mirrors the toxicity, like right along. It's a dose response.
Yeah.
And that was historically how we did things. But over the past few years, as there's more targeted drugs and more immunotherapy drugs, those toxicity curves and efficacy curves are really not one-to-one related. They are very different. And so like on the right for targeted therapies, you may be able to give a low medium dose of a targeted drug and it's highly efficacious. And so that's wonderful. But then as you think you're doing the right thing by increasing the drug, you might not notice any changes in toxicity until all of a sudden it goes to really high toxicity. And so that is a very different drug profile amongst targeted drugs and immunotherapy drugs compared to historical chemotherapy on the left. So the FDA said, wait a second, hey, now in the modern decade, we're going to have to figure out when we start doing these dose optimization, picking medium and low doses and try to optimize, we should delay that optimization choice of what the dose levels are to optimize until we get a little bit more efficacy data.
Safety.
Yeah. So the next slide says, okay, now the FDA's recent shift is to move the dose optimization decision, not the dose escalation, safety, toxicity decision, not the phase 2, declaring your phase 2 dose decision, but really what's called a dose optimization decision, which is like fine tuning the dose level until you get a little bit more efficacy. And so the good news and the bad news is the good news is this is making it so that we really can learn more about targeted drugs and immunotherapies and because they have their own efficacy, toxicity, kind of divergent curves. That's good. The hassle factor news is it makes trials a little bit more longer, burdensome, more effort in general to figure out the right dose. So it's good in the long run, but in the short run, it requires more trial infrastructure because you have to enroll more patients and be looking at different dose levels. So that's good on the one hand, but it also it's taxing to the research clinical trial infrastructure system, which is already heavily taxed. So
Right.
So it's good, but it also has kind of some drawbacks. I mean, in the long run, it's good, but doing dose optimization on all the studies that make it through phase 1, that's a lot of work. But also it's worth it in the long run. So there's still some issues that have to be solved as how is our research trial infrastructure going to handle all the dose optimization for all these phase 1s that look promising and accrue an additional 40 to 50 patients before we go into phase 3. Or our phase 3 studies are all going to be about dose optimization. Well, wait a second. If phase 3s are all about dose optimization, what's our comparator? What's the standard of which we're comparing it to? And so one of the angles with the comparators is maybe we can compare to historical control. And that's a big new thing we're trying to look at. Can we compare to we have all this great big data, right? All this historical data on how all the patients did the past few years with pancreatic cancer. So we know this historical controls. So that is kind of what's at play. If that makes sense.
It does.
But it's good for your audience to know. So this is more of a good audience question. Well, what about, this is me teaching the med oncs and the audience, well, how do we balance therapeutic intent and versus therapeutic misconception? And we talked about this before. The primary objective of phase 1 trial is to increase the dose and assess safety and toxicity. But yet in this new drug, there's no proven therapeutic benefit. Very true. However, there's the hope, there's the promise, there's the intent. And of course, the secondary objective is to determine if there is some anti-tumor effect. So therein lies some inherent conflict because physician investigators, we have the therapeutic intent to aim at the druggable target. And now in 2026, the drugs are becoming more and more targetable, unlike old-fashioned chemo, which was a sledgehammer. So now we can determine with very early efficacy signals, hey, drug hits target. They're highly engineered to hit target. You can almost bet they're going to hit target. And the target is a mutation driver of the cancer that we know, or it's an immune pathway that we know, because in the immunotherapy world, we know that. So all those things can be true, but yet despite that, it's not proven, despite that there's hope and therapeutic intent, that's not enough for a drug company. It's not enough for an insurance company to cover the trial. So there's always was this historic pushback that the insurance companies would say, hey, we're not covering this. It's experimental. We're not even going to cover the basic scans or the supportive care or the comparator arm. So we get all this pushback. And
Does that have a lot to do with like the coding when they go to look it up or?
Well, yes, in the one sense that there is no CPT code for a trial. There's what we call some qualifier codes that I think now, 10 or 15 years ago when we used to deal with this, there were some, I think, early qualifier codes. But in general, before the Affordable Care Act, which kind of declared you can't discriminate against trial patients, insurance companies would always discriminate, in my opinion, always discriminate. They always would sniff out. If you're on a trial, they would look for reasons to deny coverage.
Automatically, it's experimental.
Yeah, and we have some proof on that here in the next couple slides, and you'll see the data. So there was always this conflict that patients and a bigger deal, the insurance companies would have this misconception that, it's not proven, it's purely experimental, and the doctors would be conflicted a little bit, could say, of course it's not proven, but there's all this hope, we have all the science backing it. So that was an inherent conflict. And now with modern precision medicine and biomarkers, we can say, yeah, of course there is a misconception, there is some unknowns, but the intent, not the proof, but the intent is that drug is going to hit target. And therefore, we should bet in favor of the patient. And so this is such an important point that the two big regulatory I should say, the two big referees of our industry, right? ASCO, speaking of ASCO, and the FDA, there's the referee of the FDA, they had to step up and make these declarations. Because insurance companies were pushing back so bad past 20 years to deny patients access to trials that the ASCO and the FDA had to step up and say, here's what we're declaring, is that phase 1 trials have the potential to provide patients with therapeutic benefit. Therefore, quote unquote, phase 1 trials attempt to treat the cancer, thus offered to patients with therapeutic intent, as well as to achieve the scientific objectives of the study, right? And then the FDA comes in and the policy says they acknowledge that the primary aim of the phase 1 trial is to gain early evidence of effectiveness, just to kind of push the blowback to the insurance companies that, yeah, of course, this is not purely experimental. If it was purely experimental, we'd be researching cancer drugs on healthy college students across the street from college. That's what we would be. But we don't do that. We only experiment on college students for other drugs like toenail fungus drugs, blood pressure drugs, and antihistamine, stuff that like doesn't matter if you hit the drug target or not. I mean, I'm saying that facetiously. Of course, it matters if you have histamine allergies, but it matters a whole lot more if you're a stage 4 cancer patient and you have a mutant driver and we have a targeted drug to hit that. That is a lot more of a bigger deal, which we should not waste our valuable resources on using cancer drugs on healthy college students across the street on a phase 1 healthy volunteer clinic, which is, there's a lot of those out there. So this was, I say these things, these were recent policies of the FDA and ASCO. This is like 2020 and 2019. That's how recent we had to give the blowback to insurance companies because they were denying all the trials. Like this is like not that long ago that these quotes are coming out. Okay. So I say this, you can see the vein of my existence is to like deal with how many decades of we've had insurance companies denying trials. So how do we in the cancer clinic trenches explain to patients about this therapeutic intent misconception? Because the patients are so hopeful. We say they're vulnerable in stage 4 meta. They're vulnerable that we can't ethically take advantage of their vulnerability and promise anything, promise a cure. We can't say it's going to work, but you give them just enough hope that it's balanced. So we say, I tell them, hey, science has discovered most of the cancer drivers, check. In the immune pathways, check. Like release the brakes, step on the gas pedal in the steering wheel. And so now we are engineering drugs to hit those targeted pathways, check. You know, I can say every 9,000 cancer drugs are off. Nobody really is developing more old-fashioned chemo.
Yeah.
So I tell the patient, it's highly likely that the study drugs, they'll hit your target. But you, dear patient, you're teaching us, we as the physician investigator, are learning from you, including the sponsor and the FDA and scientific community, that if it hit targets and if it works in the side effects, you're teaching us what's the right dose, which is why there's fine tuning and why it's so dynamic. And you got to take a log diary and there's all these hassle factors. So if your body's strong enough to qualify for eligibility, we don't run out of phase 1 options. We keep recycling. And we're a rapid fire phase 1 clinic. If the trial doesn't work, we always have backups. We use scientific rationale based on your molecular profiling to choose the best trial option. And if your tumor is growing rapidly or in near obstructing or pain crisis or we're very honest with you. If you don't come in here with a support system, if you're in pain, we can't experiment on you. I mean, there's some patients I feel personally bad. We just can't. And we're honest with them. We try to give them and their local oncologist a plan, a tune-up plan, and come back with your support system. And here's these things. So that's what we explain. And I want to go to that.
Patients respect and appreciate that. That's so needed.
We do. And now we're getting better. If they're borderline, we might do a video teleconference first. So they're not driving 3 hours here.
You can assess based on a conversation before they come in. Hey, is it necessary for you to come in? They can describe their current side effects or symptoms. You can coach them through it and save them the drive or the trip. And
yeah. So here is a great reference for your audience to read in Nature Reviews of Clinical Oncology, 2019. It was a review at that time of all the phase 1 trials. What are the valid therapeutic options for cancer patients? What this is saying is to some of the insurance companies or some people that say it's not worth it, trials, it's a waste of time, they're experimenting on you, unlikely to work. Well, in 2019, they look backwards and say, what are the response rates in general for phase 1 trials. These are first in human phase 1 trials. And looking at the wheel on the clock there of these things, the response rates at that time, the response rates were overall 20% that had been reported results. That's 1 in 5% chance response rate. That's just not stability. That's like an objective overall. That's like the tumor shrinking, like an objective response rate. Next is, what about survival? For 18 to 38% overall response or objective response rate might predict an overall survival from phase 1s. That's pretty good. Safety. If you say, oh, is it safe? I'm a guinea pig. I don't want to die from a toxicity. Now, it's still an experiment on a new drug, and there can have anaphylaxis, and we are increasing the dose. Basically, dear patient, we're looking for side effects. And if you come in our clinic as an athlete and you're real strong and have stage 4 cancer, we're going to aim high. We're going to give you a high dose because you can handle side effects. But if side effects happen, the toxicity, it has been, cumulative toxicity can be 0.5 to 2%. There is a learning curve to these drugs. So that's why we have so much safety parameters around phase 1 trials, military style safety with a support system. Now, over here on the right, some other good news about the biomarkers. If you have a genomic biomarker or a genomic target like a KRAS G12D that we talked about in these, or a RAS mutation, and the overall response rate for those trials are much higher than 20%. Those trials are averaging 40%. That's pretty good. That's pretty good odds.
And RAS.
So that's just telling you that the more targeted the drug, if you have a target we can measure and detect, the more targeted the drugs, the more promising. So that's sort of why we tell patients we are looking at your molecular profile in what trials we have open to see if we can match you based on your mutation drivers into the most appropriate trial. So, and then down at the bottom, it says dosing. High doses may not always be preferable with targeted drugs. And that goes back to that comment we made earlier about that sometimes efficacy and toxicity are very different with targeted drugs. There might be a quick blip up in toxicity, and you may be able to have at lower doses have very good efficacy with less toxicity, or vice versa. So there is, it's not like chemo where it's matched toxicity and efficacy, just track right along. So this was a good review article. I think this was good for your audience to read and to learn about, if that makes sense.
Yeah, absolutely.
So next slide, what we've got, this is another big one that I told the oncologist. If you refer into a phase 1 clinic earlier, it matters. So this was an Australia-based phase 1 clinic. They looked retrospectively at their 28 phase 1 trials over 5 years, 2013, 2018, and they saw that if you referred with metastatic cancer when you were in your first or second regimen, you actually were able to enjoy the benefit of the 20 potential, overall response benefit. However, if you came in too late, where you were much more advanced, malnourished, in pain, third, fourth, fifth regimens, not only was there a higher screen fail rate, but those patients just did not respond as well. And so what that's
makes total sense.
Makes total sense. But that means to some of your audience listeners, if you're counting your regimens, not your cycles, but your regimens, you need to speak up. You need to say, hey, if I wait until I'm 5th line after 2.5 years of chemotherapy, it's harder for your immune system to mount a response. It's targeted to overcome bulky tumor burden.
Is there a term for that? T-cell exhaustion? The more beat up, the more you go through, the less likely you are to respond. You would assume that the first treatment that you're given is going to be your best option, and the more you go down the line, the less likely it is that you're going to benefit from it because you're just getting beat up.
So look at the good news, though, in this thought. If you had less than 2 regimens or were at 2 regimens and then referred into a trial, the good news is at the bottom of those patients that got referred in and passed through into screening and enrolled, of the 190 patients that were evaluable, 4% had a complete response, 18% had a partial response, which means more than a 30% decrease in the tumor burden, 31% had stable disease, meaning that the tumor stabilized.
Great.
So when you add all those together, 4% plus 18% plus 31%, that adds up to what we call the disease control rate. Did you clinically benefit? Was the tumor controlled? And that's yes, in 53, based on if you had an early referral into one of 28 phase 1 trials in Australia. Now, back in 2013 and 2018, that was when immunotherapy drugs were hot. That was probably the majority of their trials. In Australia is where there's all the sunburn. So I would bet that part of the reason their data looks good is because they were sunburned, and sunburn melanoma patients tend to do better on phase ones.
Australia is like the number one country and continent for melanoma.
Yeah, it's up there. In fact, I'm pretty sure if I remember this, but it is considered child abuse if a kid gets blistering sunburn. So it's real expected that you use the real heavy white sunburn cream in Australia. And yeah, you can't be seen with a sunburned kid as a parent.
Yes.
You can't because childhood sunburns also equate to higher risk of melanoma.
Yeah.
So moving down here to the next one. So who funds phase 1 trials? This is I'm teaching the doctors. Well, really, your medical practice funds it for at least the first 5 to 10 years because you have to build it. So you have to have quite a bit of conviction, right, to see it through. It's a lot of high overhead costs, more rent, more space, high payroll, equipment, minus 80 freezers, a lot of regular, you have people for data entry, quality, regulatory. So it's a long 10-year investment. And you have to say, is that what your community wants? Is that worth it to you to grow your oncology practice in your community? Because it'll have a longer return on investment over time. But in the beginning now, in the first 10 years, it's probably sacrifice. So the sponsors also fund it, but they're not going to fund it well enough for you to break even that first 5 to 10 years. You got to build up a portfolio, a book of business of good trials and for it to really break even. And who are you working with is you're really working with small, medium drug companies, right? Because that's where the phase 1 trial is mostly small, medium biotech companies. We do have a good diversity of large biopharma too, but most of the drugs are in smaller companies. You also have to be working with the CROs who they aren't really directly funding you, but you have to get in good business associate. You have to deliver good service to them. They're their middleman, right, who comes and audits you and tracks your site. They kind of are the go-between. So you have to give them good place to sit, good service.
Yeah.
If they ask you to do something, you better do it so that you can have a clean book of data and clean audit trail. If for us, we're lucky. My talk was to North Carolina MedOncs. Right? Hey, the CROs are here in your backyard. You got it easy because you have the middleman are all right here. They want to drive to you. Whereas you go to some desolate states where there's no research infrastructure, there's no biopharma, much less CROs. There's not a good opportunity to develop trials there. And then I mentioned here that the CROs, the contract research organizations, they select sites. We call it the official word is institutions or sites. If they have operational efficiencies, which you have to earn those. That takes a long time to have that oiled machine, have good regulatory experience and good track record of accruing, don't have too many screen failures and good accrual metrics so that you are basically showing that what you're doing, you're not having all these screen failures or getting people that don't make it to their first scan or enrolling people too advanced or too sick or God forbid don't meet the eligibility criteria. That's a big no-no. Then the FDA and the IRB gets involved if you accrue people that don't meet the eligibility criteria.
It's just good business practices.
Yeah, it's good. And then the biopharma sponsors, of course, they want you to have operational efficiency and good site metrics. But what the biopharma sponsors care the most about is investigators who are engaged. And what that means is investigators that like their domain of their interest. Like I'm an immunotherapy guy. And my partner, Dr. Gandhi, he's like small molecule guy. He likes heme, malignant heme, or brain tumors. Dr. Gandhi ran a brain tumor clinic up in Princeton for a decade. So we're actively seeking out phase 1 trials for glioblastoma. And so he has those special interests that he's engaged with, those research community trials, the patient advocacy groups. And so that matters both to the patient population who may self-refer and to the chosen sponsors who you work with. So these are little things that the MedOnc audience might not understand or appreciate because they're too busy in the clinic, but it's good to teach them. So how do phase 1 programs operate? So this is a good one. So I walked through them. This is how we set up our clinic. This is our clinical team. Just like you, we've got infusion nurses, we've got front desk, we've got medical assistants, we've got some research coordinators. We also have a pharmacy that has to have the regulatory compliance for
it's like the ABCs of your infrastructure.
Yeah, the ABC in the medical. Oh yeah, we have about half of that, or we have 60% of that. We could pivot a little bit more. We talk about how we start up, how we try to be the first site open. We try to execute through contracts and budgets, which is part of the operational side pretty quickly. You're not going to get business if a biopharma sponsor offers you a new drug and you sit on it for 4 weeks. You're not going to get business. Or if you say, hey, I want to do it, but then your team doesn't get it through contracts, budgets, negotiations, regulatory startup, you're not going to get the business. And so, unfortunately, for some big academic and bureaucratic hospital systems, some big institutions, they move like an aircraft carrier. So sometimes they have a great a great young, eager med-onc champion who wants to be a great investigator, but he can't get through the layers of the bureaucracy to open the trial. This is pretty common. And you have a moral hazard because you have patients that want it. You got a good, eager doc who's like wants to do it, but they can't get through 10 other layers. And then the drug companies are worse, the CROs. If the CROs label you as inefficient, that's harder to earn that back because the CROs depend on an efficient site for them to do good business.
Right.
So these are the things that really matter. So anyways, good teaching. And then
you
you got to
have those
be
checks and balances.
Just like most docs, you have to have good access. You got to have patients that come to you. You got to live in a demographic where there's every community has cancer. And you got to be close enough where there are some other good big hospitals in case somebody, God forbid, needs to go to an ICU or something. Our own timelines for everything we do is like a couple of days, like 1 to 2 days. We should be able to turn around everything, whether it's data entry, contracts, budgets. And I tell my docs, you got to be set up. And this is funny. So a lot of sites still are paper-based. Well, they might be EMR-based, but they're they try to do hobby. As a hobby, we're going to do some trials. It's still very paper-based. We lived with a paper-based regulatory binder and sign-offs and
files for days.
So much like training everybody and the amount of, I just had to like tell the story for one of our nurses gets married and their last name changes. I have 55 signatures because somebody's last name changes. They have to sign again on a delegation log. That means they have to sign 55, and then I have to go behind them and sign 55 times. So if there's a protocol in phase 1 trials, every protocol gets amended every few months. Why? Because it just have to get amended to open up new dose levels, new side effects happen. We have to new safety plans. So it's very dynamic, and everything you do in a regulated environment, you have to document that you trained and have proficiency and competency, and all the staff followed this. So it's a massive trust. We went through a big transition internally, going to a digital reg binder and the low-hanging
paper
fruit.
Files and you're scanning them.
Yeah. And this was beautiful. It was a hassle factor, but we did it. We got through it. And this has saved so much of my time. And then we also have gone through ways that we have further digitized a regulatory binder. And none of this is anything proprietary. This is what the FDA expects when you organize your regulatory. This is what you get inspected for. This is all public knowledge. It just shows that you have like 2 big categories of regulations, and each one of these folders has another like 25 files in it. But it's just showing these are the categories of the folders just for a specific trial on the left, like one trial. And each one of those folders has got like correspondence. Anything that we talk to the CRO or the drug company about has to go into a correspondence folder. And you can imagine 20 patients on trial for 6 to 12 months and all the correspondence, all that stuff. It can't just be in our email. It's got to be in there because it's got to be auditable and traceable. It's part of the official record. You got to be
able to track it, and it's got to be
Bingo.
seamless.
So all those forms, all the screening enrollment log is like when a new patient comes on the trial, that's like the only place we put their name in a reg binder. So we have the link to the name. Otherwise, they're known as the subject number, right? Yeah. In that particular thing. So this is the audit trail for if we get audited by a CRO or a sponsor or FDA. On the right, this is just the general regulatory, like not even trial specific, but the general regulators are all of our refrigerators and freezers and centrifuges and pipettes calibrated. Yeah. Are safety hoods calibrated? Is the humidity calibrated? Literally. And where are we keeping all of our CAPAs or corrective action plans, like root cause analysis, quality metrics, like tracking errors, non-conformances, near misses? We have to track all those very big, important things to show that we're following GCP, good clinical practices. Next, we got to keep a roster of all the ethical review, like who's on the ethical review, even though it might be a committee in California that we digitize and send the protocols to ethical review. We have to keep all those names of the committee members themselves on that as a roster that is inspectable by the FDA. They don't go to the IRB to inspect it. They can come to us to inspect it. Master delegates, even if we have a student or an intern that shadows with us and is training for like 2 months on learning about clinical research trials, or a pharmacy tech learning, and they're here for like a 2 or 3 month. They may be on a delegation log that they are trained in learning with supervision. They might actually be learning on how to do something that is part of their training. We put them on a delegation log, or just general medical assistance or things like that. They're not a coordinator that goes under the trial specific regulatory, but they are our general staff that goes under the general regulatory. We have general SOPs. We have about 10 SOPs on how to write SOPs, all where we keep our source documents. Like, is the source in the pharmacy files? Is the source in the EMR? Is the source in the, where do we keep the records of all of our temperature curves of our refrigerators? I mean, all these things. Every CV of everybody here has to be in a separate folder. Like, you don't want the FDA hunting through your HR records, right? Technically they could, but you don't want that. So instead,
You have a direct spot to go to.
when the auditors or the CROs or the monitors say, hey, you want to know we're legit here, everybody's CV, everybody's credentials, everybody's training in this big 3-ring binder, and all the temperature monitoring. So that's just the beginning of the top layer of what it takes to do trials. And so when you're telling this to an audience of med oncs that are like, maybe I might be interested in trials, but then they're like,
Be very organized.
they're like, I don't think my boss will let me do all this stuff, or my boss won't let me dedicate the resources to this. Or the hospitals may say, we don't have the bandwidth to do this. These are the real reasons why there's not that many research sites, is because that's the decision that happens when I go tout about how great research is. It's that the hospital systems that are not big enough to do it, or the medical practices that can't afford to do it, it's too much layers of the amount of work that needs to be done. And we need an industry system fix for that. So that's another talk. We're trying to make it easier with digital and trying to make it easier, but this is the way it is today. What you saw, that's the folder structure today.
It's 2026. I thought that this is the new standard now, right? I mean, you got to be organized.
That's what you get audited. If you get audited, if you do trials as a hobby, and you do one trial and you get audited, they're going to expect that you have that folder structure to rely upon your data of your patients to be submitted to the FDA. Like, that is the expectation. And so this is why a lot of docs realize, I got other data that shows how many that try it once and then stop. These are they
don't realize how much work it is to.
Yeah. So, I mean, it's important. It has to be done. It's a joy. It's our chosen discipline of practicing oncology as a research clinic. But I want to share that with you and your audience. So the audience, we tell patients, hey, man, your visits here are never going to be like fast and easy. We are a regulated environment. Everything is double checked, triple checked. Everything's a long questionnaire. Everything is just orderly, organized, and we do it as fast as we can with the constraints that we have. And we are efficient with doing all that, but it still is like many long days for patients. So this is, I think these are good overviews of what we do. This is just for screenshotting. And so your audience knows we're trying to build more informatics to make it a little bit easier to integrate these systems. Our leukapheresis for those patients that are interested. We do have like the Red Cross, we have a pheresis machine. We collect white blood cells, bags of immune cells, and we can grow them down the hallway and do practice runs to upscale them for our internal research, trying to match immune cells to tumor killing assays. So pretty cool stuff. We got accredited by the FDA for our blood bank, for our blood banking establishment to be able to do these leukapheresis as the beginning of a cell therapy starting material. So speaking of cell therapy starting material, I told the med oncs at this conference, we got to be prepared because the FDA, just like in Europe and the UK, and in Australia, they're now developing regulatory guidelines in their statutes of their countries on how to bring point of care manufacturing and even like genotype and mRNA and gene silencing, this type of regional manufacturing to have regulatory pathways to do it. I think all this is going to have to be built into research trials, but they're at least the FDA on this link here. They are laying the framework to take feedback for the guidance. I mean, this is not official FDA final rule, but it's just showing that the global industrialized nations are trying to figure out, hey, we got to deliver for our patients. We got to start figuring out how to do regional point of care manufacturing. So this is our point of care manufacturing down the hallway. That's our team really cool. Over to the right, we've got like 3 more big clean rooms, which, by the way, just passed our first true sterility microbial testing, like perfect, which was great.
All
So
right.
with all the big machines have been going for a couple of months. Everybody's been bunny suited in there, total like operating room clean. So that was a big news last week. I told their boss, I said, we got to give them a bonus.
That's cool, man.
And we got to put that on LinkedIn, man. We got to look like
Yeah.
we're making progress. So that'll be on LinkedIn and with a nice little
Oh,
celebrate. So yeah, they're not in their bunny suits there. You can't see them in their bunny suits. And they'll be
no.
behind glass. But so this was the sterile hallway that goes into the clean rooms that we have been controlled, like 45 ceiling filters to keep clean rooms. So eventually we can grow cells to the FDA specifications here that we're working on. Speaking of probes for environmental monitoring, temperature, humidity, and pressure, we have 140 probes,
Wow.
like measuring everything. Each of those probes has to be calibrated and documented battery changes and constantly 24-hour alarms going on. And you see the word SOPs down there. There's a word before that, IQ, OQ, PQ. This is like the expectations of when you have a new device coming online or new software being implemented. You have to go through installation qualified. Is it qualified to be installed? Operation. Is it operationally qualified? Have you turned it on? Is it working? Are all the clicks and buttons? And then performance qualification. Did you train your staff? Are they qualified to perform it? So we had to go through all that IQ, OQ, PQ just for our digital regulatory binder, like the Adobe signature on there. Like that was a massive 3-ring binder of just that validation process just for that. So, and you have to do that for each of the devices or each new software that we bring online that is in the clinical trial workflow. Interesting. So we have a lot of cold, I think we count like 20 freezers and refrigerators and liquid nitrogen, a backup generator. So highly regulated to be able to do the GMP in the future. I just showed that to MedOncs about, hey, this is where the industry may be for clinics and next to 10 years, and to plant that seed that maybe we'll be going them as we build our models in the next 5 to 10 years. This is just a slide to remind you that we do grow immune cells. We do leukapheresis. The patient on the left was one of our first cancer patients who donated his blood through leukapheresis, and we can grow out tumor cells right out of the bloodstream. We can count them. We can grow out immune cells. We test them against their own tumor. And then over on the right, when we take the leukapheresis product and we walk it down the hallway warm and fresh, we can bring it to the R&D. At that time, it was R&D or process development in what's a tabletop manufacturing device over on the right. That was our first large-scale run of us doing our first manufacturing batch of these immune cells a few years ago. And so we've iterated the hell out of that and made much more improvements. And that's the one we're working on now that got through the FDA for Interact meeting. So yeah, so that study, our in-house study, opened way back in 2006. So we've been doing this small
20
scale.
years.
20 years, small scale. So yeah, pretty cool. So a lot of R&D goes in down the hallway. So what are the challenges? So this goes to pivoting here. So dear oncologist, okay, so what about insurance companies? Oh yeah, this is bad. This is where it gets like, holy crap. Like this is the reality check. Like by this far, the MedOncs like, hey, Dr. Bowderly, I think clinical trial sounds really cool in theory, but I don't know if I can do all those reg binders. I don't know if I can do all that. I don't know if my hospital let me hire all those people. So then it's like, okay, let me get started on how really bad it is. Like that's nothing. Like this is the reality. So John's going top left, insurance denials for routine medical care on clinical trials. This is just not my opinion. This is the source references. Okay. Again, I love to be opinionated. I'm Irish.
No, I got you.
Don't get me started.
Yeah, I got
This is just the absolute facts that I want your audience to hear. And they should write their senators and just tell their advocacy groups we need to change on this. From 2003 to 2008, Johns Hopkins, one of the top U.S. medical research institutions doing clinical trials, found that 13% were absolutely denied due to lack of insurance coverage. Most of those were in the phase 1 trials because the insurance companies said phase 1 trials are purely experimental. There's no therapeutic intent, right? There's no therapeutic aim, right? And hence that's why 5 or 6 years ago, the FDA and ASCO had to come out with those declarations.
Yeah.
And why on the NCCN guidelines, they had to put, we recommend the best possible intent is for cancer patients to enroll in clinical trials. That was like their big box of like importance. So, but still, despite that,
We talked about this on our last couple of recordings. In fact, I was on a panel a month or two ago in Washington, D.C., and I brought up that statistic.
Yeah, it's bad. So I want your audience to screenshot the hell out of this. And Chris, you can use this as you go on your rampage.
Yeah, absolutely.
On your talks. Like, so in 2005, I won't say which oncologist, but a market dominant insurer, and I'm not allowed to say which insurer, denied in-network status to a Huntersville, North Carolina based community oncologist simply because they offered phase 1 trials. And of course, their response is, we support trials in general. We just don't support the doctors that do the trials to put them in our network, right? So, okay, go next bullet. Despite the 2010 Affordable Care Act, which was Obamacare, which had in their provisions, 2010 Affordable Care Act mandated that insurances cover the routine care costs on trials, i.e. like CAT scans. Despite that language in the Affordable Care Act, delays and denials continue to impact patient participation in trials.
Yeah.
And so here is a survey since that time to point out that a 2014 survey of not just one oncologist's opinion, but 252 cancer research centers. That's like,
what?
That is like all the cancer research centers that were community-based. That's like all the United States. And it showed on their survey that 62.7% of sites that they reported failure of insurance plans to cover trial participation as the most common reason for denials.
God. Do you have an updated statistic for 2026 compared to the 2014?
No, because to get that data, like just this 2014 data, that was a heavy lift. That was a survey that went out to all 252 research centers, asking them a 10-pager, like break it down. There has been, that was published in 2017. And on right, it actually shows what were the trials that were denied the most. And it's the phase 1 trials that were the biggest percentage of the phase 1 trials. 37% of phase 1 or tied with phase 2. That's just how bad it is. So now it's just like getting insurance companies just to cover standard of care is still a hassle. It's like, here we are crying about covering research trials, which research trials are free. The study drug is free. And we're having a hard time getting insurance companies to pay for the scans or the supportive care. Now, we also know that insurance companies in general, their reputation and the data is borne out in other data sources. I mean, make a slide deck about that. Just getting them to cover the standard is they don't like the expensive standards. That's not my opinion. This is data on that. The number of denies. In fact, almost every other night on TV, you can see these insurance denial case escalated. The local news station has to get involved in escalating. So next is, this was an ASCO. Speaking of ASCO, and ASCO did a good referee attempt to try to dig into what are the barriers to access to trials based on each stakeholder. What do they see the barriers are? And I'm not going to go through all these. This is for screenshotting and for people looking up the link. It is a good read. All these are valid reasons, and there's not going to be any one single fix. There just has to be a whole system rewrite of what is society's weight that we put into trials. How are we going to fund them as a society? Should society, just like we fund roads and infrastructure, should we start funding more trials as part of infrastructure? Those are big questions. So next is here, this is a bad one. So I'm telling oncologists at the podium, dear oncologists, there's not enough medical oncologists, principal investigators to conduct trials. Here's why. 66% of principal investigators that sign a 1572, which is their federal license to do a trial, right, only sign it one time, one and done, because they learned the hard way real fast the administrative burden. And then the sponsor, the CRO may come back to them and say, hey, this ain't a hobby. You better game up. You better get all your their first audit or second audit, because the first one they probably were told, if we're going to choose you, you better have all these things. By the time they enroll a patient or they're on to their next one, it's tough. Or this might just reflect the fact that a lot of the oncologists in hospital systems that might have interest in trials are assigned to be the administrative PI and to start signing 1572s but they don't really know what it entails until they start doing it.
And they were like, oh, God.
And they ask the hospital system, can you support me? Can you hire these 8 more people I need to do all these things? And the hospital's like, well, not this quarter.
Yeah, we don't have the budget.
And then they're like, well, maybe the FDA is going to hold me responsible because I'm the investigator. They're not going to hold the institutional responsible. They're going to hold the investigator responsible. It's the license is to the investigator to do those things, to make sure that they have those adequate time, adequate resources, the adequate oversight, adequate to do all those things. And so therein lies a conflict that maybe those young champion oncologists in academic centers realize it's hard to do. Yeah. And then of the remaining 1572s that do get filed, only 10% actually do more than 5 studies a year. So that's just telling you that really they don't stick with it. It's kind of stops. It's beyond a little more than the one and done. They might do 3 or 4, maybe 5. And then they learn within a year, this is a lot of work, not sustainable from their quality of life or whatever is the reason. So this is why most U.S. biopharma sponsors, big, medium, and small, and most CROs based in North Carolina have the right intentions. Of course, they all want to do trials in the United States, but the industry pressure and the cost advantage is pushing it overseas. It's just like all American manufacturing, like Mexico can manufacture it. These emerging countries manufacture it cheaper. I'm sure professional services are moving. I'm sure when you call to get like customer support, you're talking to another company, another country at midnight. It's just, it's all of it's getting outsourced, including the trials. So the number of PIs in the US, number of principal investigators, has decreased by 85%, the number of PIs. And so, and 1 of 5 oncologists are on the verge of retiring. Probably half my audience at this meeting going on to vacation for their conference. And there's going to be 20 million cancer survivors by 2026, a 31% increase. Cancer survivors are 26 more million by 2040. Only 2-3% participate in trials. And so this is why the U.S. biopharma industry is now running more than 80% of the trial sites ex-U.S. I think about all those KRAS studies, we are lucky we had the phase 1 for KRAS. We're now an expanded access site. But a lot of those patients only now are coming online with regional sites to get the expanded access, or a lot of those patients were international patients as well, which is great. I mean, there's many countries that we should share the wealth globally. But I also know that those are U.S. biopharma companies who were supported with NIH and NCI funding and to do some of that discovery. And maybe we should have maybe a little more U.S. trial sites. So it's like 50-50, not 80-20. This is the reality. Okay. Workforce, clinical site refer. You know what? I'll stop here because we've been going a long time. But let's get back to that because this is just the beginning of the workforce, like getting enough coordinators. And then we go into the FDA has data on how many trial sites are opening up overseas. And the FDA has data on what are the most new trials being opened. It's not the United States opening up new trials. It's China's.
Thank you so much. Real quick, I just wanted to kind of show you this because back on one of your earlier slides, you had that notebook with all the tabs. And I actually have one of the copies of my book where I have all these little extra tabs from specific notes that I always refer back to. So anyways.
Highly organized. Yes, you're highly organized.
You have to have some sort of system, digital paper. You got to be able to track it. So, all right, my man, everybody, thank you again. Sending love and peace. All right, that concludes today's episode of TIL Talks with Chris White as we talk TILs, tumor-infiltrating lymphocytes, and all advanced cell and gene therapies. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. I'd like to thank my guest today for joining me on TIL Talks. To our listeners who have not yet done so already, please follow, comment, subscribe, share, and smash that like button. For more information about TIL Talks and related resources, please visit www.tiltalks.com. I'm Chris White. As always, sending love to everybody. Peace.